The chosen report in which to analyse is the influences autophagy has on the elongation of mitochondria. The experiment and report were carried out by L. Gomes, G. Benedito and L. Scarano. Their concept, which they wanted to prove, was that when a cell is lacking nutrients needed for cell survival, it will begin to digest its own organelles in an act to gain energy to produce more. This is a form of cell recycling that this report has found to be necessary in a cell's development. However, this process has been thought to only focus on certain organelles and leave others intact. One organelle which is left is the mitochondria of the cell. In the hypothesis of this experiment, it is said that the mitochondria do not break down when the cell is starved, but rather they enlarge. The word used for such a process is autophagy. During autophagy, there are three phases, with the primary phase and most focused on being macroautophagy. Macroautophagy is where double membrane structures are formed called autophagosomes, which act as transport holes known as vesicles for the degradation of cellular organelles to take place in. Mitochondria, which do not take place in autophagy, are the energy storage organelle within the cell. From this energy storage in the mitochondria, adenosine triphosphate or ATP can be produced and then utilised for whichever energy-using task is needed to be completed. To generate the ATP needed, a process of respiration where oxygen is utilised is conducted. The result that was found from this theory is that the mitochondria are indeed increased in size, causing the cell during the cell starvation. The increase in surface area of the mitochondria cell forms a shape that is elongated, seen in figures A and D. Where over time the cells are becoming longer. The method used to prove this was culturing the cells to then starve them of nutrients by the process of diminishing the serum and amino acids. Over just 1 hour of starvation, there were signs of elongation in the mitochondria. The authors found that there is a production of protein kinase A or pKa, which is an enzyme that provides proteins with phosphate groups. So in the experiment, the pKa therefore increased the phosphorylation of proteins which maintain the structure of the mitochondria. A protein involved is dyname-related protein 1 or DRP1, which will allow for the fission of the cell. Fission is the process of dividing atoms to make a series of smaller atoms. When DRP1 has not yet been phosphorylated by protein kinase A, it will allow for fission to proceed. However, when the cell has starved and pKa phosphorylates the proteins including DRP1, the process of DRP1 allowing fusion is prevented and therefore fusion is allowed. Fusion is the process of multiple atoms fusing together to make one larger atom. Therefore, as fusion occurs, the mitochondria enlarge and elongate. Further studies in the same year by D. Arduino, A. Esteves and S. Cardoso illustrate the problems involved in the unbalanced control of fission and fusion. They found that it may be indirectly involved in the cause of Parkinson's disease.